Okay, let me first of all welcome everyone and thank you so much for joining us for our first ever virtual Susan F. Miss Center for Women's Cancer Presidential Symposium, our Breakout Center. For those of you who I don't know, I'm Jane Jameson, and it is my pleasure to serve as co-chair of this session with my friend and fellow trustee, Jane Brock-Wilson. I'd also like to extend a warm welcome both to our first-time attendees and to welcome back our long-standing participants. We are really grateful to each of you for your commitment and support of Dana-Farber and for investing your time with us today. This year's agenda was designed to reflect Dana-Farber's patient-centric approach and to demonstrate the way in which this approach informs our research and maintains the focus on bringing more effective treatments to the clinic. This afternoon, we are extremely fortunate to be joined by 8 of our experts specializing in breast and GYN oncology. We will hear updates from division chiefs and 2 panel discussions. The teams are excited to talk with you about their progress, some promising projects, and some near-term priorities. It is our hope that this year's program will highlight examples of Dana-Farber's what we like to call special sauce and underscore the substantial effort and resources, especially during these unusual times, that are needed to support the ongoing bench-to-bedside research and best-in-class care. Before we start, of course, there's always some housekeeping items. So let me just go through a few of those. We'd like everybody to feel as connected as possible, and we'd love to see and hear you, particularly as we engage in Q&A. So as you look at your screen, please find the camera and microphone icons on the top left of your screen and make sure that they are on. Michael, who's helping us with technical support today, will manage this for you and pull you on screen when you ask a question. Also, please take a minute to look at the bottom right of your screen and locate the chat and question icons. The chat icon is green, the question icon is the question mark. This is where you can submit your questions, and we will bring you on screen so that you can ask that and talk with the faculty. We do want to get to all the questions if we can, but if for some reason we can't, Suzanne and Allie will work with the faculty and make sure that we do get back to you with questions that in the event we can't get to them in today's session. So with that, let's get started. It is truly my pleasure to introduce Dr. Eric Weiner. Eric and I met, it was actually my first visit to Dana-Farber in 1997, and Eric had just started in the breast program there, and I think we can all look back and be amazed at what's been accomplished since that time. Eric is a professor of medicine at the Harvard Medical School. He holds the Thompson Chair in Breast Cancer Research. He also wears many different hats at the Institute, including the Senior VP for Medical Affairs. He serves as Chief Clinical Development Officer and also, as you know, Chief of the Division of Breast Oncology. Eric and I had a chance to say hello a few moments ago, but it's great to see you, and we're eager to hear your updates and the panel discussion. So I will turn it over to you for our first session.
Thanks. Appreciate it. I'll say to all of you that it's not without a little anxiety that all of us are approaching this to make sure that this whole virtual thing comes off. So if it's not perfect, we recognize that, and we're trying. Thank you, Jane, and thank you, Jane Brock-Wilson, for being so supportive and for chairing this today and for everything that you do, and thanks to the whole audience. Can we go to the first slide? All right, well, we can quickly skip past this. There we go. So just to give you some sense of the size of the program, and I know that many of you are familiar with this, we have over 30 breast medical oncologists. It's remarkable to me only because when I came in 1997, there were about 4 or 5. Actually, back then, Ursula Matalonis was a breast medical oncologist trying to do a little GYN cancer, and she was one of them. We have 4 radiation oncologists. We have many nurse navigators, nurse practitioners, social workers. We have 13 breast surgeons, multiple fellows, multiple research nurses, and none of this takes into account all the people in both wet and dry labs doing research. And we see truly a huge number of patients every year, and we pride ourselves on trying to do the very best by them in all situations. I want to briefly mention a few new faculty members. One of them you're going to hear from today. And the other two represent our growing international faculty. So Filipa Lins is Portuguese. She went to medical school in Portugal and trained at Georgetown, where she stayed on the faculty. And Filipa was recruited just a few months ago and is going to focus both on her own clinical work and on triple-negative breast cancer and on BRCA-related cancers. Going to the other side of the slide, Antonio Giordano is from Italy, has also received training in the United States. Most recently, both he and his wife, who's also a breast medical oncologist and who will be part of one of our satellites, have been at the Medical College of South Carolina in Charleston. And we're very happy to have Antonio, who will be focusing on drug development. And finally, and you'll hear from him later, Isaac Klein, one of our own former fellows, a physician scientist whose primary focus is in the lab, although who very much is committed to clinical care as well. I should also mention that there are 2 new surgeons who have been recruited. They're not shown here on this slide. And of course, at any given time, we have new nurse practitioners and PAs and others who are part of the program. We have a very broad clinical trials portfolio, and in truth, even during the height of the COVID pandemic, although I suppose that height may not have been the height, but even when things were at their worst, we still had clinical trials that were open. We have 100 active clinical trials. Over half of these are investigator-initiated, meaning that these ideas came from our own faculty. We have a very broad tissue banking effort, and by banking tissue, we allow for scientists to do many different kinds of experiments that lead to a better understanding of drug resistance. And we can use some of this tissue as part of preclinical models that may help us test different drugs in animal models. What we've seen over the past several years, over the past decade really, is that the increasing use of targeted therapies has clearly improved both the survival and the disease control for patients with advanced breast cancer, and has also led to more cures for women with early stage breast cancer. And you're going to be hearing more about immunotherapy, which of course is a huge treatment in cancer in general. In breast cancer, we've been a little frustrated that immunotherapy hasn't been quite as effective as it has in other malignancies. But you're going to hear from Beth Mittendorf, a surgeon scientist, about just how we think we can make that better. Do want to just make a comment about COVID. Maybe you're all sick of hearing about COVID, but we've really worked very hard to try to maintain our patient-centered clinical care and our clinical trials throughout the pandemic. In March and April, the number of visits in person declined rather dramatically, and we made up for them with a huge number of virtual visits, and we had never had virtual visits before, and suddenly we were seeing patients not just on the phone, but primarily on Zoom. To this day, about a third of our patients are being seen with Zoom visits. The other 2/3 are coming to clinic. And I think compared to this spring, what we've learned is that with appropriate distancing and safety procedures, that we can very safely see patients at Dana-Farber. And our new patient volume has actually picked up over the past several months, and we are now again seeing essentially the same number of new patients we used to. Of course, cancer doesn't wait for COVID, so it's reassuring that we're seeing the same number of patients, because if we weren't seeing them, presumably those patients wouldn't be getting care anyplace. At the beginning, there were some delays in surgeries when we were grappling with some of the problems posed by the pandemic. And I think that most of us believe that even in the face of a surge this fall, and even as cases climb, that we're going to still be able to provide the kind of health care that we need to safely and effectively take care of patients. And in truth, I think that the growth in the telemedicine world is something that we're going to take lessons from and we'll be using in the future. I will mention that we actually published guidelines for how to care for patients in the midst of the pandemic, and there were some modifications that we focused on. I'm just going to go back to this because the next slide is actually Beth's. I just want to, can we just shut off the slides for a second? You all have an ability to do that? Yeah. Just one other thing. So after 23 years, 23 wonderful years leading the breast cancer group, I'm actually stepping down from that role over the course of the next 6 months to take on some additional institutional roles, to develop clinical programs, to work on our external relationships, and to work in conjunction with our business development group as their clinical arm and their clinical voice, if you will. I think it's time after 23 years for there to be a new leader. I think change is good. I'm not going away. I'm still going to be running our very large breast cancer spore grant and some other research initiatives. I'm still going to be seeing patients. But I think it's time for the day-to-day operations, and for that matter, more than the day-to-day operations, to be taken over by someone else. And we're just in the midst of starting a search process for my replacement. And lest you think this means that I'm anywhere close to retiring, that is not true. And as evidence of that, I'm actually in the midst of an election, not as important as the election that's coming up in the United States next week. But I'm in the midst of an election running for president of the American Society of Clinical Oncology, our large professional organization. So I still feel like I have a lot to do yet. With that, I want to introduce our panel. And of course, I was given long introductions, and I'm going to make them short. So our panel consists of 3 individuals: Nancy Lin, who is a medical oncologist and is the associate chief of our division, and whose own work has focused on the treatment of patients with brain metastases and research, specifically clinical trials that focus on patients with brain metastases. This is a huge unmet need for patients with metastatic breast cancer, and Nancy is, in my view, the world's expert in this area, and has been working in this field for now well over a decade. Second, you will hear from Beth Mittendorf, who is a surgeon and is the director of our breast immuno-oncology program. Sometimes we think of surgeons as being less focused on systemic therapies, but Beth is an example of someone who is an outstanding surgeon and at the same time both a laboratory scientist and a clinical scientist. And finally, you're going to hear from Isaac Klein, who's a young, although he might argue not so young anymore, but a young medical oncologist. Isaac is a physician scientist as well. His primary focus, as I mentioned earlier, is in the laboratory, and he's in a laboratory at MIT and hopes to have his own laboratory sometime very soon. But Isaac, who worked with me as a fellow, is now seeing patients independently in the clinic and is an outstanding doctor in addition to being a rising star in his work in the laboratory. So I think we're going to now move to our panelists, and we're going to start with Nancy. I recognize that the theme today is optimization of care, and we're going to ask each of our speakers to weave in how what they're doing relates to optimizing care. And finally, I'll just close my remarks with one last comment, which is that in the world of breast cancer, we're we are pursuing two separate paths in terms of research. One is we're asking for all the women who are going to be cured, how can we minimize therapy as much as possible and as safely as possible to limit late side effects and early side effects and still achieve the same long-term results? And there's a huge effort going on in this area, and we have been at the forefront of it. And at the same time, we know that there are some patients for whom we just can't do enough with our present therapies. And we're looking to develop new and better therapies. It doesn't necessarily mean more aggressive therapies or harsher therapies. It may mean smarter therapies. And so that is very much a focus of ours as well. So I'm going to start with Nancy, and Nancy's going to talk just a little bit about brain metastases. Nancy.
Thanks. So brain metastases refers to breast cancer or any kind of cancer that spreads to the brain. And in patients who have early stage breast cancer, fortunately, this is relatively rare. But for patients who have metastatic breast cancer, it is actually distressingly common. So for patients who have HER2 positive metastatic breast cancer, about half of patients ultimately over time will develop brain metastases, and a similar proportion in patients with triple negative metastatic disease. Historically, the only treatments we had available were radiation and surgery, which, as you can imagine, have potential side effects associated with them, particularly if we're talking about radiation to the entire brain, which for many years and decades was really one of the most effective and only treatments we had available. And it's an area where medical oncologists in general have shied away from research. In fact, patients with brain metastases until relatively recently were not allowed to enroll in virtually any breast cancer systemic treatment trial. So many years ago, Eric actually was one of the authors of a paper with a former fellow from the Dana-Farber, really describing this phenomenon for the first time, that patients with HER2 positive disease are at very high risk. And that really led to this whole area of research in trying to develop better treatments for these patients. And so I'm happy to say that over time we have developed treatments that at least have some degree of effectiveness. The most recent treatment was just FDA approved, and it's called tucatinib. And it's a pill treatment that targets HER2, gets into the brain, and it's actually the first treatment that's been shown to improve the survival of patients with breast cancer brain metastases, the first systemic treatment to show that effect. That took many years to get to that point, and still patients are not cured of their metastatic breast cancer nor of their cancer spread to the brain. But I think it's a very positive step because it shows that with dedicated research efforts that we can really make a difference in people's lives. And just to give you a sense of the scale of improvement, at the 1-year mark in this particular clinical trial, 70% of patients who had brain metastases were alive in the tucatinib group versus about 40% of patients in the other group. So, I mean, this is a very big difference just thinking about such a short-term outcome of 1-year survival.
Thanks.
I don't want to go too long, so I mean, I can talk for a long time.
How about just a few sentences on the challenge of taking care of patients with brain metastases, which is something that you do more than anybody else?
Yeah, so I think that this is, I think there's several challenges. One is just the emotional challenge, because of course people are sick, and they, and the brain is where, people's function comes from, and it's very scary to be somebody dealing with a diagnosis of brain metastasis. It's also complicated because it requires, to be able to take the best care of patients, just as we do for early stage breast cancer. We're not the only game in town, i.e. medical oncology. And so we really do have to work very carefully with radiation oncology, neurosurgery, and neuro-oncology colleagues. And so we have built a program to really formalize that collaboration that we've done all along, but to really make it even that much more robust and stronger. So we have a dedicated group of specialists within each of these subspecialties that actually take part in our breast cancer brain metastasis program. And that really allows us to, every time there's a treatment decision to be made, we really are able to, again, in the sort of spirit of personalizing and individualizing treatment, that we can come up with a plan that makes sense for that specific patient. And we, have the good fortune of being able to work with such great neurosurgeons, such great brain-focused radiation oncologists and neurology-trained neuro-oncologists. And, that really helps us take care of each patient the way we really want to and the way we should. And I think that is not the case in many places.
Thanks. All right. Well, we're going to turn to Beth. Let me just say that probably the smartest thing I ever did in my job as the breast program leader was to recruit two outstanding world-class breast surgeons, Terry King and Beth Mittendorf. And it is something that I will forever both take credit having participated in and be enormously proud of. And from a immunotherapy standpoint, Beth has transformed what we have going on, and she's going to speak to you about immunology and immunotherapy in breast cancer.
Thanks, Eric. That was very kind of you. Michael, if I could ask you to pull back up the slides. I do have two slides to show. And as Eric and Nancy and Isaac know, I could talk about, go ahead and go to the next slide. I could talk about immunotherapy all day, but before I do that, I actually wanted to make two comments, Eric, to get back to what you were saying about COVID. I don't think you gave yourself enough credit for the work that you did with Terry King, the chief of breast surgery, and Jennifer Bellin, the chief of radiation oncology, to pull us together as a team to think about how we could thoughtfully care for patients during this crisis. And I personally was incredibly proud to be part of that team and felt that I was able to offer my patients the same level of, or an outstanding level of care that is not going to in any way compromise their oncologic outcomes. And so thank you for those efforts. The other thing I would say about COVID, you mentioned that our volumes are back up. I would suggest there's one area where they're not quite back up yet. As a surgeon, we tend to see the very earliest stages of breast cancer, ductal carcinoma in situ being an example. And most women with DCIS, they're diagnosed through a screening mammogram, and we still aren't quite back up to our volumes for screening mammograms. And I just want to encourage everybody that both the Brigham and Dana-Farber, we have created incredibly safe environments. I feel quite comfortable here. I feel comfortable with my family here. And I would encourage folks to make sure they get their screening. With that said, I did want to talk a little bit about immunotherapy. And you can see the title of my slide here, Harnessing the Immune System to Fight Breast Cancer. I got involved or interested in immunotherapy about almost 15, actually probably longer than that, maybe 18 years ago now. And at the time, a lot of my mentors were naysayers. They didn't think that there was ever going to be a way to harness the immune system to fight cancer. And in fact, we now know that that is exactly what we probably need to be doing. And I still believe that by the time I retire, we're going to figure out a way to treat every breast cancer patient with some form of therapy that enhances their immune response and their ability to fight their tumor. This is a slide that I like with a couple cartoons on it. On the left, what you're seeing in the purple, that's a single tumor cell. And the little gray circles, those are T cells that are recognizing that tumor as foreign. And that's exactly what T cells do. We all have T cells floating around in our peripheral blood. These are the kind of the soldiers of the immune system that recognize when something's foreign. And when they do see it as foreign, they're able to attack it and destroy it. And so clearly we would want for our immune system to think of cancer as being foreign. And in fact, it does. The issue is once the T cells, those little gray circles, recognize that purple circle as being foreign and begin to attack it, they actually put on brakes. And the reason they put on brakes is because that for a typical immune response, such as to the flu, or if you were to scratch yourself with a rusty nail, you wouldn't want that immune response to go in perpetuity. Like you do not want it to go out of control. Whereas if your immune response is against your breast cancer, you probably want it to go and you want it to go full speed. And so what I'm showing then on the right is a figure that understanding this figure is what has allowed us to figure out how to therapeutically treat patients by augmenting their immune response. So you see there the big gray thing that says T cell, and on the left-hand side, there's things called activating receptors. So if you think about an activating receptor, that's like stepping on the gas pedal. So if you step on the gas pedal, you're going to get those T cells to recognize your cancer. They're going to attack and destroy it. As I said, though, once T cells get activated, they put on their own brakes. And those are the inhibitory receptors shown on the other side of that figure, the CTLA4, the PD-1. And that's effectively like stepping on the brakes. But of course, if your immune response is against your cancer, you do not want those brakes activated. And in fact, there's a whole class of drugs now that are being investigated that take the brakes off, that allow the immune system to go. So if you go to the next slide, as Eric alluded to, we didn't have initial successes in breast cancer. The initial successes for stimulating the immune system to fight tumors were seen in melanoma and lung cancer. But in breast cancer, in the last 18 months, we have had what I kind of refer to as a victory. So patients with metastatic triple-negative breast cancer, they were enrolled onto a trial that was called the Impassion 130. And what that study did was it took nearly 1,000 women and it randomized them. So half the women got chemotherapy plus a form of immunotherapy. And the immunotherapy was a drug that takes the brakes off, so allows those T cells to go. And the other half received chemotherapy and what's referred to as a placebo. And what I'm showing on the right, we call this a Kaplan-Meier curve, but this is looking at what was the length of survival for these patients in the two arms of the trial. So the women who got the placebo, they're reflected in the red line. And the way to interpret this is that their survival was about 15 and a half months. If we added the immunotherapy, those are the patients shown in the blue line. And what we see here is that their survival was 25 months. So a 10-month improvement in the survival of women with metastatic triple-negative breast cancer was eye-opening to all of us who take care of breast cancer patients. This is very clinically significant. And based on these data, as well as other endpoints from that trial, this drug, atezolizumab, again, one of the drugs that takes the brakes off of the T cells, has now been approved for use in patients with metastatic triple-negative breast cancer as a first-line treatment. So when Eric, Nancy, and Isaac see patients with metastatic triple-negative breast cancer, they can test their tumor for a marker to see if, in fact, their immune system might be susceptible to being augmented with this drug. So what's really exciting about this is that in the last year, we've had another trial that looks at a similar patient population and used another form of immunotherapy. And we see in that trial as well an improvement in survival. And so we suspect there'll be a second agent approved here in the coming months. And then perhaps of great interest to me as a surgeon, we found that if you now move these drugs that have been shown to work in patients with metastatic disease to the earlier stages, so give them to them before I would take them to the operating room. We've recently heard from 2 different trials that, in fact, if we give these drugs with chemotherapy before we operate on them, when I do their surgery and the pathologist looks at the tumor and the lymph nodes under the microscope, we are more likely to have found that the drugs, the chemotherapy and the immunotherapy has completely eliminated the tumor. And we call that a pathologic complete response. And we know that patients who respond to therapy by having a pathologic complete response have better outcomes. So although we were not the first, breast cancer was not the first to show benefit of immunotherapy, I think we're well positioned now to build on the successes we've seen in the last 18 months. And as Eric alluded to, with the basic immunologists, the translational researchers, and the clinical trialists we have here at Dana-Farber, I anticipate that we're going to be one of the groups that helps to lead the way.
Great. Thank you, Beth. And finally, Isaac. Isaac, do you want to just share something about your own work and how your work in the clinic also enhances what you do in the laboratory?
Sure. Thanks so much for having me. My apologies for the mask. I'm in between experiments now. So as Eric said, I spend most of my time in the lab, but also see patients in the breast oncology clinic. And my research I consider my research quite basic, but I try to be inspired by my patients and the need for further therapies when thinking about what kinds of questions to ask in the lab. And so that what that brings to my research, I really think is extremely valuable. My work in the lab is really focused on understanding the cellular processes that will be the target therapies that I think will be forthcoming in the next 5, 10, or 15 years. And one of the major challenges in figuring out how to target proteins that are really important for breast cancer and really all cancer growth is that lots of those proteins don't form these very nice structures that we can easily design small molecules for so that they can bind and inhibit those proteins. And something like 40% of the proteins in a cancer cell fit this description, where medicinal chemists can't really design small molecules to bind them effectively. And there are lots of very important breast cancer oncogenes that fit this description. And what we understand now from the research that we've been doing in the lab is that these proteins with these non-structured regions form compartments in the cell that we call condensates. And these compartments have special physical properties and chemical properties that we're just starting to dissect. And what we've recently discovered is that we can actually find small molecules that are and small molecules that can potentially be developed into drugs that can actually find these compartments with these heretofore undruggable or untargetable proteins, concentrate inside them, and disrupt their function. And so, to the theme of optimizing care, my efforts are really on, when Nancy and Beth figure out how to, utilize the current therapies to the best of our abilities, and in 5 years we're asking what's the next step for those patients. That's what I think about. I think about how we're going to develop the next generation of drugs, and how can we understand the processes that are going on in the breast cancer cell that will be the target of those drugs. And just to add one other word about what kind of environment this is, as you might imagine, it can be hard to convince people that time spent in the lab, hunched over a bench, understanding what we now call condensate biology is relevant for breast cancer treatment. And I think it is, and being surrounded by people like Eric and Nancy and Beth and everyone here who can be inspired by that kind of vision and understand that motivation is really a special privilege.
Questions. I don't see any in the chat room, and I'm going to pose questions. I'm going to first make a comment, though, which is that I think a lot of the success that we've seen, and obviously we want to have far greater success in the future, is because we all recognize that it takes a village, but more than that, it actually takes a cohesive village. And that's something that I think we in our program and we at Dana-Farber and Dana-Farber and the Brigham have been able to do that over the past many years. And I think it's so very critical. So I'm going to ask each of you one question. Since our time is limited, I'm gonna ask you to try to keep the answers relatively brief. So Nancy, having brain metastases sounds terrible to anyone. Where are we in terms of being able to prevent brain metastases? Can we do that in anyone? Do our therapies that we give to patients with early stage disease help prevent brain metastases?
It's a great question, and it's a very timely question. If we look at people who have early stage HER2 positive breast cancer, we cure the majority of those patients. But if we look at a recent clinical trial, which is called the Catherine trial, it took high risk early stage HER2 positive patients, and patients received standard chemotherapy, Herceptin, and then they received a medicine called TDM1 after surgery. Most patients are cured. But of the patients who are not cured, of the patients who relapse, half of them have cancer in the brain as the first place the cancer shows up. And that used to not happen because we used to be so bad at controlling the potential for relapse in the liver or the lung that the brain was just a really uncommon place. And that's changed. So again, fortunately, most people are cured, but the ones who are not are increasingly having this problem with brain metastases. And in that trial with the PD-1 treatment, which again substantially reduced the risk of cancer showing back up in the liver, there's no impact on the chance of cancer showing up again in the brain. But there are clinical trials that are being designed and that should open over the next year that Dana-Farber investigators were very heavily involved in designing, including a study where patients receive a HER2 tyrosine kinase inhibitor, in this case tucatinib, the same one I was telling you about before, who are going to be receiving it in the early stage setting. And it's for two purposes. One, to see whether it further reduces the risk of recurrence and increases the cure rate in general as a whole. But it's also a really important question about whether it has any impact on the development of brain metastases. And we're hopeful that it might, because if we actually look at the data in the metastatic breast cancer setting from the clinical trial that led to the approval of tucatinib, we actually saw that not only did it extend survival, but it also lengthened the time pretty substantially for patients to develop either new brain lesions or to develop worsening of their existing lesions. So it did seem to have an effect on prevention as well. It's not a definitive trial to ask that question. You have to look at it in the early stage setting, but I think it is hopeful. And then the other part of it is that in collaboration with laboratory investigators, so Shahryar Kabraji and Nelly Polyak and Jean Zhao, we're trying to understand which genes actually are responsible for the ability of breast cancer to get to the brain. And there's a lot of experiments in the lab right now going on trying to understand that, because if we can actually figure that out, then we could design smarter clinical trials that are not just based on let's try this because it's a drug that works in the brain, but actually let's try this because we actually think there's a critical need for a certain gene or gene pathway in order for breast cancer to get there. And we might be able to be smarter about that. And we also might be able to be more targeted about it so that we can find those patients who have, for example, cancers with that specific pathway that's gone wrong. And those are the patients, if we knew that those were the patients who were at risk for brain metastases, then we could actually give the extra treatment just to the patients at risk and spare everybody else sort of extra side effects. So I think we are getting there. We're not there yet, but we might, in a couple of years, I think we might potentially have results of a trial in the early stage setting that shows a prevention effect in brain metastasis, which would be very exciting.
Great. Well, I actually was keeping time incorrectly, and we have 12 more minutes, so we're going to be able to do a little bit more. So 12 minutes is a lot. So Beth, one of the challenges with immunotherapy is that it can have side effects, and specifically it can have side effects because sometimes when you unleash your immune system, of course, it fails to recognize that we are not the enemy. Can you talk a little bit about that? And can you also speculate as to whether it might be possible to prevent those kinds of problems in the future?
That's a great point. So I don't know if how I described it, this came across, but if you think about immunotherapy, we're treating the immune system. We're not necessarily treating the tumor. And to your point, the immune system recognizes, it does what it's supposed to do. It recognizes inflammation or abnormal cells anywhere. And so for folks on the call, any of the itises, so you can get colitis, pneumonitis, hypophysitis, all the itises, and that's because you get an immune response somewhere. And so I think there's a couple of important points. One, the medical oncologists who are administering these drugs have gotten much better at recognizing and treating these side effects. So that's an important point. Second, we're beginning to get some data that suggests which ones of these drugs have more side effects than others. So we'll probably take that into consideration when we think about which of the immunotherapies to administer to our patients. Third, in an effort that we are initiating here at Dana-Farber, we're going to start collecting tissues from patients treated with immunotherapy. And the hope is, much like Nancy just described for brain metastases, can we learn something about the genes in the tumor that might help us identify who's going to have toxicities so that we can do what you just suggested, which is preempt them. So perhaps pre-treat the patients in order to prevent the toxicities. And then the last point I'd make on this subject, because I highlighted my enthusiasm for bringing these therapies into earlier disease settings, we as a community have to be quite thoughtful about introducing therapy that has toxicity associated with it to our earlier stage curable patients. So in that population, we have another opportunity to, again, look for two things: markers of who is actually going to have that pathologic complete response, therefore really need these additional treatments, and two, who might have toxicities. So your point is well taken, and there's a lot of ongoing work.
A different sort of question. What about the challenge of getting funding? You know, you're there in the lab. It's challenging to support that. And, you know, what are the next steps for you in terms of advancing your career by obtaining funding?
He's on mute.
You're on mute, I think.
Oh.
Although it doesn't, I don't.
You're good now.
Am I good now? Yeah. I'm not sure what you're talking about, Eric. I've never had any problem attracting money. Kidding. So especially, I missed the very first part of your question, but I understand you're just asking about the difficulty of a physician scientist at this stage of one's career getting funding to continue work. And there's two issues. One is that the classic funding mechanisms address very clinically oriented science and very basic science. And there are actually very few sources of funding for people like me who are interested in the very earliest stages of translation, taking basic science understanding and discoveries and starting to move them to a place where we can think about applying them to cancer patients in need. And that's an issue I think that applies to people across career stages, and one that I think if you ask anyone here, they'll tell you how difficult that can be. But of course, at this very early career stage, when you're just starting to establish your own ideas and your own approaches and methods, finding the time and finding the money to initiate that work can be very challenging. I have to say, though, that I have no complaints, because Eric and Dana-Farber, really the whole institute, because I think they really understand the value of folks with a clinical understanding doing the basic science and guiding those questions, have really allowed me the time, the money, and the focus to do this kind of work. So the support the institute provides for this kind of approach is just, the importance of it can't be overstated.
I realize that that's all entirely self-serving, since we recognize that you're incredibly intelligent, creative, and hardworking, and you're the best investment that we could make. So I said that we weren't going to talk about what's often called de-escalating or optimizing therapy, meaning backing off in some areas. But we have a few minutes, and I thought we would touch on that as another very important topic. And I thought maybe first, Beth could make some comments about how much progress has been made in terms of surgery and our ability to back off on really not all surgery, because we still need the surgeons and patients still need the surgeons, but on the extent of surgery.
Yeah, so it's interesting to me as a surgeon that the medical oncologists over the last couple of years have become really excited about, we say de-escalating. I like to think of it as right-sizing therapy. Because in surgery, well before I got in the field, I mean, I have a few gray hairs, but I wasn't here when the standard treatment for all women with surgery, I wasn't suggesting anything, where all women with surgery had to have what was called a radical mastectomy, where we removed the breast, the underlying chest wall muscle, and all the lymph nodes. And clinical trials were done that demonstrated that, in fact, you didn't need to do a radical mastectomy. We could do what was called a modified radical mastectomy, where we just remove the breast and the lymph nodes, but leave the chest wall muscle. A subsequent trial suggested that, in fact, we don't have to remove the breast for every patient. So patients with small tumors, or patients with tumors that you guys make small with giving the chemotherapy first, we can do a lumpectomy and combine that with radiation. And so what I tell my patients is that all of the data we have suggests that lumpectomy plus radiation equals mastectomy with respect to my ability to cure you of your cancer. And in fact, we've now learned that for certain populations, we might be able to do the lumpectomy alone without the radiation. And I alluded to Jennifer Bellin, one of our colleagues in radiation oncology, and she's helping to lead a study where we're looking at, getting back to this theme, the genes in an individual patient's tumor to see if they're favorable, in which case I, as a surgeon, can just do a lumpectomy and then we could omit the radiation. And so we've been de-escalating therapy and surgery for a really long time now with respect to the breast. And we've also been doing it with respect to the lymph nodes. So again, when I started in this field, which wasn't all that long ago, every woman with breast cancer had all their lymph nodes removed. And we now learn that we can, in fact, do what's called a sentinel node biopsy. And so that helps in that it identifies the high-risk lymph node, but spares the patient the morbidity associated with removing all the lymph nodes, which if you ever Google lymphedema and you see a big swollen arm, we don't see that often anymore. And so, we've de-escalated a lot in surgery, and we're actually looking forward to, as you all figure out the best targeted therapies, to be able to potentially, as you suggest, omit surgery altogether.
Walk the earth, or when surgeons performed radical mastectomies. So it's not, it was a while ago.
Radical mastectomies were a while ago. Modified radical mastectomies.
Oh, no. During the course of my career, I have seen tremendous advances in surgery in terms of the ability to right-size the surgery for the patient and the tumor.
Yeah.
And maybe in the last couple of minutes, we could talk about this in relation to systemic therapy. Nancy, can you just comment briefly about what we've learned about estrogen receptor positive cancers and our ability to back off there on therapy? You're on mute.
Okay. As recently as 2000, the National Cancer Institute of the United States basically made a position statement that every woman with a diagnosis of breast cancer more than 1 centimeter should be offered chemotherapy. And the truth is that's a lot of people being recommended chemotherapy, in many cases for very marginal benefit. So there was a very important set of advances that use different molecular tests. One of them is called Oncotype, there's another called Mammoprint, but they function on the same idea, which is that can we actually look at the activity level of particular genes, and does that activity level actually predict the likelihood that chemotherapy will be beneficial or not? And also to look to see what is the value of endocrine therapy, and is endocrine therapy alone going to be an effective treatment? And with the advent of these tests, we've seen the use of chemotherapy go down dramatically, but at the same time not compromising patient outcomes. If anything, we actually improve patient outcomes, because if you think about it, if on average a patient might get 2 or 3% benefit from chemotherapy, because that's kind of what it is if you give all people more than 1 centimeter of chemotherapy, that means that many patients are getting chemotherapy for 0 benefit, and some patients are getting chemotherapy that's saving their lives, but you can't tell the difference. And so what these tests really help us do is to distinguish in which patients is chemotherapy potentially life-saving, and in which patients is chemotherapy only going to add side effects and actually have no impact on patient outcomes. So we've really been in the vanguard of actually developing clinical studies and involvement in clinical studies, and then actually translating the results of those studies into clinical practice, which ultimately is that last really important step, because you can have really amazing studies, but if nobody actually pays attention to them, it doesn't matter. And I think this is where, Eric and Hal Burstein and others in our group, have very influential voices and have been able to really change clinical practice, both in the United States and internationally, to really reduce the overuse of chemotherapy. And again, the point is not that we're giving up anything. It's actually the opposite, where we're actually not giving up any effectiveness, and we're just giving up the side effects. And on the flip side, it also means that when we counsel patients to recommend chemotherapy, that we can be that much more confident that there is a positive or favorable risk-benefit ratio. So when somebody's going through the side effects of chemotherapy, which is not easy, that you can have more confidence that actually these kind of side effects, which we obviously want to ameliorate, are ultimately going to be worth it in the end. And I think that to have a little bit more knowledge about this has made such a tremendous impact on patient care, again, in a way that's really focused on what can we do to maximize the benefits of treatment and minimize the unnecessary risks.
Thanks. No, and of course this it's both the personality of the cancer and the amount of cancer and the preferences of the patient that we put all together to come up with our recommendations.
Yeah.
So I want to thank all of you for your comments and your participation in the panel. And I'm now going to introduce my colleague, Ursula Madalonis. So as I mentioned, many years ago, Ursula was a breast cancer doctor, but she was someone who always knew that she wanted to do something a little different. And she really founded the GYN oncology program, certainly the medical oncology part of that program. And what Ursula has built in the past 15 to 20 years has been truly spectacular. And she now has, as you'll hear, a large group of clinicians and investigators who she works closely with. She and her colleagues were recently awarded a very large grant, and it's a real pleasure to introduce Ursula to you, who is Chief of the Division of Gynecologic Oncology and Professor of Medicine at Harvard Medical School. And I turn this over to you, Ursula. And you're on mute.
Am I off mute?
Yeah.
Because my little button says I'm not on mute, so there clearly is another way of muting us besides us self-muting ourselves. Anyway, it's a pleasure to be here today, and I just want to congratulate Eric on doing so much for the breast program, creating the breast program here. And Eric is on my personal. It's not the speed dial anymore. It's like a speed text. He's on my speed text, so I'm always calling upon him for advice. So thank you, Eric, so much. So I'm here today to talk about our division, give some divisional updates, just a few minutes, and then introduce our, I think, really fantastic panel. And I definitely want to see some questions. I want to see some questions popping into this chat box. So please send some questions. Can we pull up the first slide? Oh, that's not my slide. All right, there I am. So I'm gonna do this myself. So this year has been, I think, truly a remarkable one in so many different ways. I am so proud of our Division of Gynecologic Oncology, and that's all of these members. I'm proud of our oncologists, our surgeons, all of our research team, our nurses, the folks who answer the phones. This is a really special time for COVID. And I think we really cannot, there's an impact on patient care, and we see that every day, where we have to mask up and gown up. Patients are not allowed to bring visitors in unless they have some extenuating circumstances. But there's really been a profound impact on our faculty in terms of, we're not able to see each other. So we have Zoom meetings. We have Zoom meetings once a week, Thursdays between 3 and 4. And now we include the nurse practitioners. So that's our one time as a group to talk about patients, potentially special cases, special patients, issues that are evolving on our clinical floors, research that opportunities that are happening, research that has been reported out, how we're changing patient care. So that's our one opportunity to really see each other, as opposed to seeing each other in the hallway and bumping into each other, which is so normal. I meet with faculty at least once a week, and sometimes more than once a week, and we really talk about their families, how COVID has impacted them personally. A lot of faculty have young children, and it's really been hard. It's not been an easy time at all, and it's again really a testament to our team that we're holding together. And it's really been quite a productive year, and I think we're going to hear about that our 25 minutes continues on. Our division has grown to over 70 members, and I see everybody because we have a monthly GYN Oncology town meeting, and in fact, it's this week. At what point I present kind of high-level overview about our division, what's going on in our division, what's happening within the institute, because I think it's hard for most folks, like our nurse navigators. We have 7 or 8 great nurse navigators who answer our phones. We don't physically see them anymore because they're at home. A lot of our clinical research team is at home. Our new patient coordinators are at home. So it's really a time, and I make sure everyone's turning their camera on so I can see everybody. Our goals remain to really provide outstanding clinical care to our patients, make an impact on improving care through clinical, translational, and basic research, expanding preclinical platforms. And you're going to hear about that from Alan D'Andrea and Joyce Liu today about how those expanding clinical platforms or basic science platforms and translational platforms have allowed us to get a grant like the SPORE grant and how we're going to use them clinically. And a real focus on faculty development and promotion. Promotion not just through the Harvard Medical School system, which is notoriously arduous, but also through opportunities and really making sure that faculty are thriving. We have 2 new faculty, one of whom you're going to hear about today, and that's Betsy Lee. Betsy is one of our former fellows. She just started in September, and she's been seeing patients. She's been writing clinical trials. She's been writing grants. And she's also our liaison to the new Center for Cancer Therapeutic Innovation, which is our new phase 1 group led by Lena Gandhi. Rebecca Porter is also one of our former fellows at Dana-Farber MGH program. And she's been sequestered in the lab at MGH, but has, because of really long-standing interests, clinical interests that have formulated with Joyce Liu, she has now entered our team and is seeing patients, but also actively in research. And our clinical trial queue has moved through, and through the efforts of Joyce Liu in her role as the director of clinical research, we have been able to keep our clinical trials open. We even have a clinical trial that requires hospitalization to the Brigham, and that trial continued despite having the COVID surge in March, April, and May. So Joyce has done a fantastic job. And we've really been able to open clinical trials in all of the GYN cancers, and that was one of our goals a few years ago. And you'll hear from Betsy today about ovarian cancer, and she's working on clear cell ovarian cancer and writing a clinical trial. And then also from Joyce Liu around endometrial cancer, which is a focus of ours starting a few years ago, where we realized we had personally no clinical trials in endometrial cancer. But also there were very, very few nationally. So she'll talk about her endometrial serous cancer, a trial that she wrote of a drug called the WEE1 inhibitor with really strikingly positive results, and is now moving to a registration study that's been supported by a drug company. Alexi Wright works on outcomes research, and we also have a lot of banking trials. And something that I think our faculty are great around is that we're even though we've got a large group, not as large as the breast group, but we're not competitive with each other. There's lots of research to do within GYN, but everybody is really helping each other out, mentoring everybody. Joyce and Panos, as senior leaders within our group, are mentoring the junior faculty, and that's really been wonderful to see. I'm going to let Alan D'Andrea really talk about the SPORE grant, but this was just awarded a few weeks ago in August of 2020. We have multiple collaborating institutions as are here. Principal investigators are listed in addition to Dave Spriggs, who is at Mass General and has been a great collaborator in this grant. And the projects are here all around ovarian cancer and really hard to treat ovarian cancer situations like PARP inhibitor resistance ovarian cancer, harnessing the immune system, which is in Project 2, using a neoantigen vaccine. And Project 3 focuses on a combination of drugs that is being used for a particularly hard ovarian cancer population to treat who have these cancers are RAS mutated. So I'm going to pop the slides down. I'm going to move to this next slide, but then why don't we drop the slides? And I really want to introduce Alan D'Andrea, who's the director of the Center for DNA Damage and Repair. He's also the director of the Susan F. Smith Center for Women's Cancers, and he is internationally known, widely known, for his work, groundbreaking work on DNA damage and repair. And it's really been through Alan's efforts that the basic science side of the spore has really been lifted, and it's been such a wonderful opportunity to work with Alan. It's just great. I'm truly honored. Betsy Lee, another participant in the panel today, is a new member, the newest member of the Division of GU Oncology, and she also, as I mentioned, serves as our new Phase 1 liaison, and she's working on clinical trial writing for rare gynecologic cancers. And Joyce Liu is integral to our group. She's the director of clinical research, is now a senior leader within the division, actively mentoring a lot of faculty, and has led numerous trials, phase 3 studies, phase 2 trials, phase 1 studies, and really has an expertise in writing clinical trials. So she's going to talk about some of her exciting work in endometrial cancer. So I'm going to turn to Alan. And Alan, thank you for being here and welcome. Can you talk about the spore and what it means for ovarian cancer, for patients, for us, for the Institute?
Sure. No, very, very happy to do that. Ursula, thanks for that nice introduction. And thanks, obviously, to everyone in the audience for attending our presidential symposium. So many of you know me. I actually am a basic scientist now, but I trained as a pediatric oncologist, and I spent many years studying childhood leukemia and genetic diseases. And about 15 years ago, I had a very unexpected eureka moment where I realized that I was studying this rare disease called Fanconi anemia, but the defect in these unfortunate children that were born with this disease is very similar to a defect that's observed in high-grade serous ovarian cancer. So I had a crisis at this moment in my career because I had never taken care of a patient with ovarian cancer. I knew very little about it clinically, but I was very fortunate to team up with Ursula and Joyce Liu and Panos Konstantinopoulos and others who've been teaching me about the disease clinically. Now, we have, again, the purpose of the symposium today is optimizing care. So I wanted to talk a little bit about how the basic discoveries from our laboratory and from the unexpected, work that came out of childhood leukemia can now be used to optimize care. And I think that the spore is really much an example of that. So as Ursula referred to, the spore grant has 3 projects. The first project is combining 2 drugs, an ATR inhibitor, which I'll tell you about in a second, with a PARP inhibitor for the treatment of PARP inhibitor-resistant cancer. The second project, which is largely generated by Panos Konstantinopoulos and Kathy Wu at Dana-Farber, really focuses on personalized medicine, actually generating a vaccine which can be used in combination with so-called immune checkpoint blockade to treat individual patients with ovarian cancer. And then the third project, which I'll say very little about because it's quite complex and in fact not even my expertise, I'm learning a lot from it, is combining a drug called trametinib and navitoclax, which is really a collaboration between Ursula and Joan Brugge at Harvard Medical School. So we have these 3 projects, we have cores, and we were successful in raising this $12 million ovarian cancer spore grant, which has been very exciting. And these spore grants, you have to understand, are really a team effort. We need clinicians, we need basic scientists, we need translational investigators, but we need young investigators, we need we need pharmaceutical colleagues and collaborators, and we also need advocates. Advocates play a very critical role our advocates actually attend all of our SPORE meetings very religiously. Now, let me just say a couple things about the first project in the SPORE, because I think to me it's particularly interesting. So PARP inhibitors were identified probably 15 years ago now as a treatment for high-grade serous ovarian cancer. And they were a breakthrough drug. Until PARP inhibitors came along, really we were relying entirely on platinum and taxanes for the treatment of the disease. So the PARP inhibitors were a breakthrough drug. And we were able to identify individual patients with ovarian cancer whose tumors might respond to a PARP inhibitor because those tumors had a so-called predictive biomarker that told us that that tumor was most likely to respond to a PARP inhibitor. In fact, the predictive biomarker came largely from a study of the BRCA genes and the study of the Fanconi anemia genes, which again I'd worked on earlier in my career. So this was very exciting. A new drug, and it's an oral drug, and it's a relatively well-tolerated drug. There are some problems with the effect on the bone marrow, but by and large, this drug can be taken over a long period of time. And it was very exciting to see ovarian cancer patients who were responding to this drug and when their tumors were regressing. But as Ursula referred to, the big problem with PARP inhibitor treatment now is tumors become resistant to the drug. Very frustrating. The tumor may respond over a period of 10 or 12 months, but then the tumor starts to grow again. So we went back to the laboratory and we started to study the different mechanisms, molecular mechanisms by which a tumor could become resistant to a PARP inhibitor. And tumors are unfortunately brilliant. They figure out ways to get around our drugs. So for instance, the tumor might undergo a reversion and correct the genetic mutation, correct the biomarker so the tumor no longer responds to the PARP inhibitor. Or the tumor might figure out a way to pump out the drug so that we try to get the drug into the tumor cell, but the tumor pumps the drug back out again. But one of the things that turned out to be very interesting was that the tumor, in the process of becoming resistant to this PARP inhibitor, was now upregulating a new protein in the cell, a protein called the ATR, the ataxia-telangiectasia related protein, or the ATR protein. And when the tumor upregulates this protein, the tumor has figured out a way to reverse the PARP inhibitor activity. So this was an extremely exciting recognition because new companies were now generating small molecule inhibitors of the ATR kinase. And we realized that these PARP inhibitor resistant cancers had a vulnerability. That's what we're always looking for in these cancers. We're looking for a vulnerability. And what we did, we and others now, is we started to combine an ATR inhibitor with a PARP inhibitor. And these are exactly the kinds of clinical trials now which are so critical to our ovarian cancer SPORE grant. And we do this in very interesting ways. And I'll just say a couple things about this before turning to the other panelists, is we use model systems. So for instance, we'll take a biopsy from a patient's tumor and we'll implant it in a mouse and we'll generate a colony of mice which carry this particular patient's ovarian cancer. And now we can perform a clinical trial on the mice, on the colony of mice. And some of those tumors might respond to a PARP inhibitor, some might respond to the ATR inhibitor, but the tumor in these mice responds particularly well to the combination of the PARP inhibitor with the ATR inhibitor. So this is our way to actually conduct a clinical trial on a colony of mice with the same drugs that are being used to treat the patient from which the biopsy was derived. So we can do a so-called co-clinical trial. And actually more recently now, we've turned to so-called organoid cultures as a model system, where a patient who has ovarian cancer, we could actually get a biopsy of the tumor right from the operating room, working closely with the surgeon and the pathologist to identify the tumor. We get the biopsy of the tumor, bring it back to the laboratory, mince it up into a single cell suspension, put it in Jello, or so-called matrigel, and we grow the tumor into these little organoid, these little microtumors. And now we can do drug testing on these little microtumors. So this is even faster than the mouse models. And it's the kind of thing that we can actually generate information quickly, give it back to Ursula, give it back to Joyce, and maybe that we look for a correlation between how these little organoid tumors are responding and how the patient's responding. So model systems are a really important component of our spore grain. I could tell you about this for hours. I'd love to have people come visit when the pandemic's over. Why don't we move over to the other panelists first, Ursula?
Yeah, Ellen, that's great. Thank you so much. And it is there's like one slide or two slides in this, but it's taken about 4 or 5 years of work to get to this point. So so congratulations. I want to turn to the next slide here, which is another kind of a way to look at the spore. And clearly, part of the SPORE grant is that middle aisle right there that says career enhancement. And that's something that our group, with help from Alan and from others, has really focused on. So really fostering junior faculty development. Betsy, I'm going to turn to you to talk about a little bit about you, to introduce you to our group. And then your new role in the new Phase 1 Center, the CCTI, which I can't always remember what it stands for, but we always call it CCTI now. And then you also have a role, and I neglected to mention this before, but in the new BRCA Center that Panos Panjan Chowdhury and Judy Garber are leading.
Yes, well, thank you so much, everyone, for having me here today. It's definitely my pleasure to be able to tell you about my role in the Gyn-Onc division. So as Ursula did mention, I was a fellow here, did my fellowship at Dana-Farber and Mass General, and recently just began as an instructor, where primarily I see patients within the Gynecologic Oncology division. Part of my work is also to see patients within the CCTI, which is the Center for Cancer Therapeutics Innovation, a little bit of a long name, but the CCTI for short. And that center is really a combination of the early drug development center, so early phase trials, combined with the Center for Immuno-Oncology. And the CCTI will now serve as essentially a home for all of these early phase trials as well as immunotherapy-based trials. The CCTI primarily is essentially a repository for trials that may, for example, be focused on one particular mutation, using a drug for targeting one particular gene, but may be applicable to many different tumor types. For example, one drug, one gene mutation that we can see in both lung cancers and GI cancers, and maybe even gynecologic cancers. And my role within the CCTI is to take a look through the very large portfolio that exists of different clinical trials, pull out the ones that are applicable to our gynecologic oncology patients, for ovarian, for endometrial, cervical cancers, and bring them to our group and say, if there is a patient who is in need of a clinical trial, if there isn't something within the gynecologic portfolio, then I can offer this through the CCTI. This is a new position. The CCTI is a new center. I think something that is very important is to maintain continuity of care for our gyn-onc patients. And so as a medical gynecologic oncologist, hopefully that will provide a level of continuity, even though they will be seen within the CCTI. I'll have a foot essentially in both the Gyn-Onc clinic and the CCTI clinic, have a foot in each of the portfolios of clinical trials. And in that way, sort of having this very broad overview, being able to find the best, most rational therapy or trial for that patient that I'll be seeing.
Yeah, and I think, the Phase 1 program has been so critical to us as a group. Jeff Shapiro, who formerly led the program, but really still has such a big part in it, has been so instrumental in a number of our faculty gaining access to trials through the National Cancer Institute. And he's still with us, and obviously we're working with him very closely around those studies. But Betsy now is our new liaison. I think we're really excited about kind of this next step for our patients, and really having a card-carrying GYN oncology medical oncologist in the phase one center. So thank you, Betsy. And I'm hoping more questions are going to come in, but expect some more comments from me. So I'm going to turn now to Joyce, and if you could pull up the last slide, which would be terrific. As I mentioned, our group a number of years ago recognized the importance of this cancer, endometrial cancer, and it really, and Joyce can go over these points, but I'm just going to make 2 points. One point is that of the two cancers that have falling survival rates, so worsening survival rates in the United States, are both gyn cancers, endometrial cancer and cervical cancer. And secondly, for endometrial cancer, there's a significant disparities issue here, that Black women are diagnosed at later stages than white women and have poor 5-year survival rates. So we have a lot of work to do because this cancer, the number of cases per year is increasing. So Joyce, can you talk about, Joyce has been such an integral part of our program in many of her roles. So many roles, many hats. One hat is the Director of Clinical Research. She leads one of the cores on the SPORE, the organoids core. And then thirdly, Joyce also is our external satellite representative, really helping to guide care at our Dana-Farber satellites. So Joyce, talk to us about endometrial cancer and what you have been doing with this cancer.
Thank you so much, Ursula. So I think, it's really been interesting to me, sort of as I've grown as a clinician and as an investigator of these years through this group, that endometrial cancer was never something we had sort of on our radar when I first started. It was sort of another cancer, but most women tend to have early stage cancer, and we thought about it that way. And I think as I treated more and more women, it became increasingly apparent, as you can see from the statistics of this slide, that actually endometrial cancer is a big problem for us. That while many women are diagnosed with early stage cancers, there are many women who are not. And as Ursula alluded to, endometrial cancer is one of the few cancers in the US where both the incidence and the mortality is actually rising. And in fact, when we talk about ovarian cancer, for many years, we've talked about ovarian cancer as the 5th leading cause of cancer death for women in the US. Endometrial cancer is actually the 6th leading cause. So it's not actually very far behind at all. And Ursula mentioned this earlier. We really recognized this a few years ago as a program, that we didn't have trials and we weren't doing research to advance care for these women, and we need to do that. This is a long-standing challenge and really a growing challenge in the GYN space. And in addition to the fact that we didn't have trials, it turns out that for endometrial cancer as a whole, we don't actually have a ton of treatment options. So until a couple of years ago, from a chemotherapy perspective, when we had somebody with a newly diagnosed sort of advanced endometrial cancer, or one that had just come back, we would give them chemotherapy with carboplatin and paclitaxel. And that was sort of the therapy that we knew had the highest likelihood of working. But if the cancer came back or grew after that initial treatment, actually the options left are very limited. Some patients may respond to a hormonal pill, but many of them don't. And traditional chemotherapy response rates are about 10 to 15% in endometrial cancer that has grown after carboplatin and paclitaxel. In the breast panel earlier, there was discussion of immunotherapy, and immunotherapy works for some women with endometrial cancer, but actually unfortunately, a lot of our patients don't respond to immunotherapy either. And all of this goes to illustrate that this is an area where we have a true unmet need. We need new and we need effective therapies that will really help our women with endometrial cancer. And so as we were developing these programs, one of the projects that we've put together is a project of a new targeted therapy with a drug that's called a WEE1 inhibitor. And it's one that I think I'm certainly very excited about. I think our group is very excited about as a potential treatment that could one day become a new standard therapy for certain types of endometrial cancer. One of the reasons I think this project is pretty neat is it builds on the lessons we've learned about targeted therapies from ovarian cancer. And it's allowing us to expand those ideas to other types of cancers. So Alan was talking about PARP inhibitors earlier. And, I think many of you here can remember how we've talked over the years about PARP inhibitors and ovarian cancer. And it seems so routine now for us to think about how they work and that we're targeting DNA repair. But I actually remember when I was a fellow and these papers first came out, like this was a totally novel idea that we could take this drug and attack this weakness in DNA repair that certain ovarian cancers have and use this as an effective way of treating these cancers. And as we've pioneered the use of PARP inhibitors in ovarian cancer and other cancers here at the Dana-Farber, and with the support of many of you who are listening to this, I think one of the things that we've also grown, and Alan was talking about this as well, is we have a greater understanding of why these drugs work. In our process of learning about PARP inhibitors, we've learned about DNA repair and DNA replication, the process by which DNA duplicates, and how important it is to the cancer cell and how we can target it so that cancer cells actually die when we target it, and what are the features that make cancer cells more vulnerable to it. And it was because of this work that we've done that we identified that there was a particular type of endometrial cancer called uterine serous cancers that actually have many characteristics that suggested they would be vulnerable to this drug that's a WEE1 inhibitor. And this is a type of drug that actually specifically targets DNA replication. Uterine serous cancers are this tricky type of endometrial cancer. I've mentioned already that endometrial cancers are hard to treat, but uterine serous cancers can be even harder. They're aggressive. They come back even when they're diagnosed at an early stage. There are some studies that have estimated that up to 40% of the deaths that occur from endometrial cancer are actually from uterine serous cancers. But it turns out that, as I was mentioning, some of these cancers actually probably have this Achilles heel. And what we see when we look at the DNA of these cancers, when we've sequenced all of the DNA, is that a lot of them have these changes in their DNA that make them really dependent on the safety brakes that are in place in every cell, so that when it's dividing, it's able to make sure that its DNA is repaired correctly and that its DNA is being duplicated, replicated correctly. And these safety brakes are proteins in the cell like WEE1 or like ATR, which Alan was talking about earlier and is part of our ovarian spore project. And when we take the safety brakes off in a uterine serous cancer cell by blocking WEE1, for example, with the WEE1 inhibitor, suddenly what happens is these cancers can't repair and duplicate their DNA correctly, and they don't know it. So when they divide, they're dividing in a state when their DNA is not ready for division, and they undergo what's called mitotic catastrophe. Essentially, their DNA kind of falls apart, and this causes the cancer cells to die. So because of what we saw and because of these characteristics that we saw in uterine serous cancers, we started a study here at the Dana-Farber to test whether a WEE1 inhibitor could shrink uterine serous cancers. And I'll be really honest, this is a trial that nobody really wanted to do outside of us. The company that made a WEE1 inhibitor was like, well, I guess you guys could do it if you really want to. And we were like, we really want to. And so this trial ultimately, was really supported by philanthropic funds. This trial would not have happened without philanthropic support. And we actually were really proud of this trial. I mentioned earlier that when these cancers come back after initial treatment, the likelihood of other chemotherapies working is maybe 10 to 15%. But in our study, when we treated women with these cancers with a WEE1 inhibitor, we saw that 29% of the women who went on the trial, the cancers actually shrank. And in fact, if we looked at the trial as a whole, about 50%, about half of these women had cancers that didn't grow for at least 6 months. So in the uterine serous cancer space, this is really exciting. We've presented these results at a couple of large meetings. We have a publication that we've submitted and hopefully will get out very soon. And this has led actually to the development of additional trials that we hope will ultimately lead to advancing this treatment into a new therapy. Ursula, I think you're on mute again.
Someone there's like a mute elf. It's really exciting, and I think Joyce says it so matter-of-factly, but it's been a lot of work. Convincing the company to give you the drug and got a little bit of funding, and running this to the end, and then having the results come out as spectacularly as they do. And then, moving on to a company-sponsored trial where you're the international PI of the study. So it's super exciting. Betsy, and I'm going to go to Betsy, then I'm going to go to Alan. Betsy, you're a brand new faculty. You just started. You started in the middle of COVID. Just tell the group, because I have to think back to my memory banks, and I must admit I can't remember when I first started as a faculty member. What's it like seeing patients, as a brand new attending, and obviously you've got the COVID overlay issues as well.
It's certainly quite a change, but I have to say that I've had excellent mentorship, primarily from Joyce as well as from Panos Constantinopoulos during my fellowship. I really felt welcome to the group. One of the things that really drew me to staying here is how collegial everyone is, how cooperative the entire group is, and really sort of the depth of research, both clinical as well as translational. And I do have to say that trying to get clinical trials off the ground, trying to conduct translational research based on patient-derived samples, whether it be tumor biopsies or blood samples, it takes a very special set of skills that aren't easily learned and requires a lot of guidance, a lot of mentorship, which I have certainly felt from the group since starting as an instructor. Going back to that trial that you mentioned of the clear cell ovarian cancer, that is something that I've been working on with a lot of guidance from Joyce. And it has been quite a learning experience, but something that's very motivating to me, certainly wanting to not only offer a better therapy for women with clear cell ovarian cancer, but also really looking at biomarkers or features of their tumor, features that we can pick up perhaps in peripheral blood samples that may inform on whether there are specific features that show us why they either may or may not respond to treatment. And certainly this whole process of trying to get a clinical trial written and off the ground and getting funding for it has taken quite a bit of legwork, a lot of mentorship and guidance and patience from Joyce and from yourself as well. But starting as a new faculty, even during COVID, has been more seamless than I would have thought.
Well, that's good. I'm glad to hear that. Thank you. Alan, question for you. We are using PARP inhibitors really a lot in our patients with high-grade serous ovarian cancer. So these are the type of ovarian cancer which has underlying DNA repair problems and most sensitive to a PARP inhibitor. But unfortunately, most of our patients will recur. You talked about the SPORE project of the PARP inhibitor plus the ATR inhibitor. Are there other strategies for treating women who have PARP inhibitor resistant cancer that you're excited about?
Yes, no, there definitely are. And by the way, I really love Joyce's description of safety breaks, and how I thought that was such a great metaphor, that these tumors that become PARP inhibitor resistant, or they become resistant to other chemotherapy, and this could be either ovarian cancer or endometrial cancer, that these tumors are really smart, unfortunately, and they figure out a way to slow down and repair their DNA and repair their damage. So when Joyce cuts the safety brake with her WEE1 inhibitor, those cancer cells start growing like mad, and now they're vulnerable to other drugs. So I mean, this safety brake idea is just such an important metaphor for what we're doing with ATR inhibitors and WEE1 inhibitors. But yeah, we're looking for other ways to make to resensitize tumors. You know, as I kind of said, PARP inhibitors are important drugs, oral, well tolerated. They combine well with other agents. So Panos Konstantinopoulos and Nina Farkila just published a paper from our group on combining a PARP inhibitor with immunotherapy, and that combination resulted in a 18 to 20% positive response in patients who really had a very, very small chance of responding to a single drug. So that's one way. Also, we can combine a PARP inhibitor with a PI3 kinase inhibitor. That's work that Ursula published last year in JAMA Oncology. And these are important combinations that we're trying. And these are not just random combinations, by the way. There's a real molecular rationale for these combinations. That's why we study these model systems. If we can work out a combination in a model system, like a mouse model or an organoid model, and see real synergy of these two drugs, now we can move forward with the clinical trial. Actually, just as importantly, when we do the clinical trial, we want to collect biopsies from the tumor, from the patients, before and after the drug combination, in order to do studies on those biopsies and try to identify the reason that some of these patients responded to the drug combination, and just as importantly, why some of these patients did not respond. So I think coming up with novel combinations on the basis of rational scientific principles, and then testing those combinations in clinical trials, is the best way to move forward with overcoming drug resistance.
Yeah, sounds good. Agreed. Joyce, I'm really building on Alan's point around development of preclinical models. And when we say preclinical model, meaning testing combinations or new drugs within a mouse, and that mouse can have an immune system, it may not have an immune system, and you have been really integral and key in developing many model systems. Just a few comments from you about opportunities and about challenges of these different model systems.
Yeah, I mean, I think, going back to many years to when I was a fellow, and Ronnie Dratkin and I worked together to initially establish our mouse models. And we've really, I think it's been so demonstrative that good model systems can teach you so much, because we established this panel of 14 models, and we've been able to leverage them to discover new combinations, to validate combinations that we see in cell cultures. These have underlaid, the ability to get clinical trials through the NCI that they've approved, because we've managed to show that there's evidence that these work together. There are weaknesses to mouse models. They're expensive. They take time. And this is where the organoid models are really exciting, where they are things that can potentially be established more quickly, and that they may be able to, we may be able to get answers more quickly, and also with a smaller number of cells. The things that we have to establish is that these reflect what we see in the mice. And I think that's work that Alan and the Organoid Biobank that we're working together on, we're trying to establish. Weaknesses in these models also are they don't have immune systems. The organoids Alan has been developing a way that we can look at the immune response in the organoids. But in the mice, we don't have immune systems, but there are ways to make that happen. And that's something that, as you know, we're working together to build. And we also have genetically engineered mouse models. All of these are pieces of the puzzle that we have to pull together to get at the answer, because every model has a couple of weaknesses, but has real strengths that allow us to understand the biology of the therapies we're testing.
Yep, thanks so much. And on the realm of immunotherapy, which we have talked a little bit about today, at least one of the SPORE projects is around a neoantigen vaccine. I want to turn to Betsy now, and I know we only have a few more minutes. I'm going to ask Betsy a question, and then I'm going to ask you guys for some closing thoughts. Betsy, you have an interest two ways on immunotherapy. One is trying to figure out for patients with endometrial cancer, which patients might respond or not respond to immunotherapy. And then secondly, you have a clinical trial, which I mentioned before, on clear cell ovarian cancer, looking at the combination of immunotherapy and a drug called lenvatinib, which is an oral medication that blocks new blood vessel growth. Quick comments about those projects?
So the first project that you mentioned, I'm looking at not only actually the endometrial cancer patients, but really all gynecologic cancer patients who have received immunotherapy, looking at whether they have responded or have not, and looking further back to see what specific molecular changes, what genetic changes they have, and whether those are changes that are associated with either response or not response. And I think that is something that will be informative and will add certainly to our overall understanding of why immunotherapy may not work as well as we want it to, specifically within gyn cancers. In regards to the clinical trial, so that is trying to use a specific immunotherapy called pembrolizumab and pairing it with a small molecule inhibitor called lenvatinib, which it has multiple effects, but primarily it's used to block blood vessel growth. It not only blocks blood vessel growth, but also causes some changes, mostly within the tumor microenvironment, that could promote a more inflammatory response and thereby try to make the pembrolizumab more effective. There are very specific molecular features of clear cell ovarian cancer that predispose to us using this specific combination, and that is something that we are very hopeful for.
Sounds good. Okay, I'm going to ask you kind of some closing thoughts. So just tell me Betsy, what you're excited about and what your hopes are for this coming year.
So what I'm excited about is to really delve into the world of clinical trials and to really parlay our use of patient samples, whether it's blood samples or tumor samples, bring it back to the lab, and then using those results to drive forward with more clinical trial ideas. Learning more about the clinical trial process is also very important in order to be a successful trialist. And my hope is that my work in the CCTI will also give me those skills and to bring better care to our gynecologic cancer patients.
That's great. Thank you so much. Joyce, a question, and then I'm going to ask you the same question. So the first question it comes from the audience, and it's around, do you know why the incidence and the new cases per year of endometrial cancer is actually going up? Thoughts around that?
So there are some hypotheses. I think that some of it is related to our general sort of body habituses, that there is a driver for endometrial cancer growth through increased estrogen. And even the more aggressive cancers, which we usually think about as not being necessarily as responsive to any hormonal therapy, it seems like they are still driven in some form in terms of how they're occurring by estrogen, because those are among the ones that are rising in terms of incidence. And then in terms of sort of my hopes for the coming year, I think, I guess I think about it more in terms of like for GYN cancers as a whole, if I get to change your question around, Ursula.
I guess you can, it's okay.
But I think that, one of the things that really struck me in thinking about optimizing care for patients was in the breast panel, there was talk about, being able to right size care and to reduce the amount of things we do. And, I would love to be in that position one day, but I think our GYN cancers, unfortunately, women die of these cancers, and these cancers frequently come back, and we're not in a position yet to decrease what we do. It's to figure out what works for who, and to figure out, when we think about our targeted therapies, our PARP inhibitors, our ATR inhibitors, our combination therapies, our immunotherapies, which don't really work in ovarian cancer, but one day we hope to make work, that we can identify who needs which therapies. And that really requires going back to the biology and the science, taking patient tissue samples and collecting them. And I think about that as our mission sort of on the academic side, because that's where this science is being done. It's, the large and discrete trials are great in terms of, okay, we can validate that a drug has activity, but it's in the academic world and our labs here, in the collaboration with the basic scientists and our translational scientists, that we are actually making the discoveries that will allow us to truly optimize the care, such that we're thinking about a particular therapy for patients with particular types of cancer.
Sounds great. And there are actually some questions that are coming in. So I'm going to ask you before Alan, I'm going to ask Alan a question, but I'm going to ask you one quick question, because I want to make sure we're, and I'm going to, so it'll be yes or no answer. It'll be like when Hal Burstein does his speed questions at the end of the masterclass. Okay. Are there any trials looking at, this is for Joyce, are there any trials looking at preventing ovarian cancer recurrence?
Yes.
Perfect. Okay, good. Right. Alan, so your parting thoughts around goals and aspirations and what you're excited about. And there is one question for you, and it's for non-uterine serous cancers. Are there other DNA repair drugs being tested like ATR in ovarian cancer? I know that may not be a fair question. And Joyce,
what excites me the most is early detection. We don't even talk about it. We need to talk about it. And actually, Ursula and I and David Spriggs, we're building a new project in our SPORE grant for early detection. There's no substitute for early detection. I mean, if we can identify patients at risk genetically who are likely to get ovarian cancer, or come up with a blood test to determine that we got an early cancer here, let's do something about it. That's the best treatment, is to detect early. But until we can do that, now you heard all our talk, we're just trying to overcome drug resistance by putting in combinations of drugs. But there's no substitute for identifying the tumor early. And the answer to our participants' question is yes. There's a number of DNA repair inhibitors which are working their way into the clinic. We had a PARP inhibitor, now ATR inhibitor. You heard about from Joyce the WEE1 inhibitor. There's a called polymerase theta inhibitor, which came out of my own laboratory. So there's a bunch of DNA repair inhibitors that are working their way into gynecologic cancer treatment.
Awesome. Thank you guys so much. It was a great panel, and thank you for your time and your insight and your hard work. So thank you so much. I want to move on to introduce Jane Brock Wilson. I have the honor of holding the Jane Brock Wilson Family Chair. I see Jane's name all the time when I sign my emails, when I look at the chair in my office at Dana-Farber. So Jane Brock Wilson, you are like in my mind all the time. So again, I am indebted to you, and thank you so much. And please finish off this great session.
Thank you, Ursula, and I apologize for the long last name. That definitely doesn't help, I'm sure, for you. I'm sort of the other Jane in this. It's so great to work with Jane Jameson, and we appreciate this. It's totally been a marathon today. I don't know about you guys. We all do a lot of Zoom type calls. This is long to go from 9 to 3, so thank everybody for your attention. Ursula, Alan, congratulations on the SPORE. That is crazy, prestigious, speaks to the merits, the promise of your work. To all our panelists, it's wonderful to see all the respect you have for each other. We also know all the camaraderie that exists between all of you. It's hard to see that on these types of calls, but it's so vital to what you do every day, and we totally appreciate it. COVID-19 has obviously drastically changed all of our lives. It's been interesting for me to hear the impact it's had on patients, families, providers, as well as the delivery of care and research progress. So thank you it's obviously on our minds with the news all the time, and it's great to hear how Dana-Farber has been coping with it. I don't see any more questions that have come up. But if you have one, throw it on quickly and we will pick it up. Thank you in the meantime to all the faculty members for joining us today, for the life-saving work that you do. I personally learned something today from each one of you. There was some takeaway. So I think we all value that, everybody that's on the phone and have hung in here with us. Eric, we're really excited for you and your next chapter at Dana-Farber. We hope you'll join us at future meetings and update us on the new ways your work advances Dana-Farber. Good luck with the elections. I've already done my vote for next week, but if I could vote for you, I would do that as well. So we all wish you luck in that. On behalf of Dana-Farber and the patients they serve, I'd like to underscore our gratitude to all the attendees who are here today for your support, your commitment in advancing cancer medicines. We appreciate everything that you all do. We understand your time is precious, and we appreciate that you chose to join us for a pretty long day today. So I know we will next hear closing remarks from Dr. Glemsher. So please stay safe. Don't delay your own health screenings. I know we say that to everyone else, but we need to do that ourselves. Get your flu shots. And we hope we can see each other in person in 2021. So I think that covers us because I'm not seeing new questions come up. So I think if IT can pull up the closing remarks from Lori, if that works, that would be great.
Hello again. I hope you all enjoyed a productive breakout session and the time to connect with one another. Please accept my sincere gratitude for your participation. And again, I would like to thank Dana-Farber's trustee and faculty chairs whose names you'll see on your screen for leading today's discussions. They have diligently taken note of your questions and ideas, and I'm looking forward to meeting with them next week for an executive session to ensure that I am apprised of your questions and your ideas and your feedback. While, of course, we would have preferred to meet in person, as we have for nearly 2 decades for this symposium, one distinct opportunity of this year's format is the ability to expand access to this program to an even wider community. In fact, at the executive session last year, one of the main recommendations from our trustee and faculty co-chairs, based on your feedback, was to share this symposium more widely through video and technology to enable even greater participation for those who might not be able to get to Boston. To that end, recordings from today will be available to you and to those who couldn't join us live. As well, the community and resources sections at the top of this event platform will continue to be available in the months ahead, and those are additional ways to stay engaged with each other and to spread the word to others about Dana-Farber's advances in research and care. At Dana-Farber, together, our community is a force to be reckoned with, and we can't ignore cancer. Our together is comprised of you, our foremost advocates, and our physicians, nurses, clinical support staff, whose compassion is a beacon of hope, and our scientists, who are among the most brilliant minds in the world, and children and adults with cancer, who motivate all of us to accelerate prevention, therapies, and cures. Our together is embodied by the Jimmy Fund and our mantra, we're all Jimmy. We're all Jimmy, where Jimmy is every patient, doctor, nurse, staff member, donor, and event participant, everyone who joins to support groundbreaking cancer research and care at Dana-Farber, as you will witness on the screen now to close today's program. I look forward to continuing this conversation. Thank you so much for joining us, and I wish you strength, wellness, and peace to you and your families.
Jimmy is a boy, saved from leukemia by Dr. Sydney Farber. Jimmy is a girl, a nephew, a husband, a wife. Jimmy is every patient ever at Dana-Farber Cancer Institute. Their families, friends, neighbors. Every doctor, nurse, and researcher at Dana-Farber. Every event walker, runner, rider, and golfer. Support the Jimmy Fund and give more and more of us a second chance at life. Because we're all Jimmy.